Hi everyone, I'm Amal H. I am the Director of the Food Allergy Social Education Center of Excellence at Cincinnati Children's Hospital. I've called the talk SOP, SOW, SOS for FAE and the FCNs, and you'll pretty soon know why. The talk is about our Discover Together Biobank and Biomarkers, which is a grant that we got from FAIR with FAIR 2.0 initiative. Here's my team. It's a large team. My co-PI is Dr. Macaluso. He is the Director of Biostatistics and Epidemiology. My co-Is are Mike Pacello, who directs the Discover Together Biobank, James Morgan, who manages the biomedical informatics. Then I have several co-PIs from the Division of Allergy Immunology and the Division of Biomedical Informatics, and as well as the Division of Microbiology. I also have two young faculty trainees, Dr. Justin Schwartz and Ashley Devonshire, and we have amazing support personnel in our coordinators and our analysts from the James Anderson Center. So what's a biobank? A biobank is a biorepository that accepts, processes, stores, and distributes biospecimens and associated data for use in research and also in clinical care. Biobanks evolved from being small, predominantly university-based repositories for specific projects or diseases to becoming institutional and government-supported repositories. The NIH does support a whole lot of biobanks. Biobanks have been adopted by commercial and for-profit organizations, store and then distribute or sell the samples. The biobanks have also evolved into population-based biobanks. For example, the UK Biobank is a population-based biobank, and virtual biobanks that people can access from anywhere around the world. The data itself has also progressed from being just basic data of the date and time of collection of a sample and the diagnosis to gathering more extensive information about the patient phenotype and the sample processing metadata. And eventually now the samples or the biobanks keep extending into genetics, proteomics, and all other forms of omics. The purpose of the FAIR Biobank is to be a disease-specific biobank. It's supposed to provide tactical and logistical services for the management of food allergy-related biospecimens and data, establish guidance on biospecimen collection and handling, and work closely with the FAIR Data Commons to ensure linkage to other critical data elements, such as the medical history, treatment, and outcomes for each patient and sample. I'll tell you a little bit about our biobank, which we call the Discover Together Biobank. It was founded in 2007 and houses about 1.2 million samples and related data from over 160,000 patients. It houses the samples, the data, the infrastructure to support the institutional research as well as external customers. It provides biobanking infrastructure to support investigator-initiated studies, sample acquisition, processing, storage, and distribution. The Discover Biobank has several available sample collections already there. The one that's currently enrolling is what we call the Discover Together, and it is a targeted, prospective, dedicated blood draw collection. We do consent the participants for sharing, for full medical record use, and even the permission to recontact them for future studies. The previous version of sample collection was the Better Outcomes for Children protocol, which really used the residual blood collected so we can use it for DNA analysis. And still the participants there were consented for biorepository projects. The collection that really started the biobank is the genomic control cohort, which for now has about over 1,000 control sample populations representative of the Cincinnati metro area. And it is collected from schools and normal individuals who have no disorder, so they can be used for genomic controls. The Discover Together Biobank supports multiple networks, and some examples of these are the Genomic and Research Innovation Network, which is known as GRIN, the March of Dimes Prematurity Research Center, which is the Ohio Collaborative, the Down Syndrome Achieves, known as DSA, and more recently, the Electronic Medical Records and Genomics Network, which is known as eMERGE. Moving from Biobank to Biomarkers, we need the SOPs. And why do we need that? Because we need to decrease pre-analytical variables, which are any variations that take place between the moment the specimen is collected and the moment of specimen analysis. The three factors that may have an effect. The first is the physiologic variables during the collection, the biospecimen processing variables, and the influence or interference factors. This is why SOPs are necessary and standardized, and our biobank has a collection of standard operating procedures so that any sample that comes in or leaves the biobank is covered by the standard operating procedures. The purpose of the FAIR Biomarker Discovery Center, which was the second part of FAIR's ask, is to have a biomarker discovery center, is to identify disease-specific biomarkers and their significance in diagnosis, prevention, treatment, and management, and to collaborate and coordinate with the FCNs, with some non-FCN sites and FAIR partners for food allergy-specific biomarker discovery and clinical research. And this is where SOWs come from, which is statement of work, which we had to submit to FAIR to say, well, this is all we can do for FAIR and for the FCNs and for even non-FCN sites to participate in the process of biomarker discovery. There's a couple of slides here that go over what we offer at Cincinnati Children's as biomarker discovery centers, which are basically the research cores at Cincinnati Children's that extend from bioinformatic collaborative services and cores to clinical mass spectroscopy facilities, confocal imaging core, data management and analysis centers, DNA sequencing, gene expression cores, imaging centers, MAG cores, microbial genomics and metagenomic labs and media labs, as well as nuclear magnetic resonance-based metabolomic cores facilities, the pathology research core, pluripotent cell facility research flow cytometry core, the transgenic animal and genomic editing core, translational trials and development support lab, the vector production facility, veterinary services, viral vector cores, and all that is supported by our Office of Clinical Trials and Translational Research. So this is my rendition of what the FAIR 2.0 FCNs is, which is starting with the already established patient registry, which is contributed to by the FAIR members. Last I heard has 12,000 participants at least. The registry then will be managed by the data commons. The patients in the registry, a lot of them come from the clinical care centers of the FCNs, which in turn contribute to the clinical research centers and the discovery centers of the FCN 2.0. The patient registry could also be contributing to the biobank and biomarkers discovery centers, and this is what we are going to be working on. At the same time, there'll be contributions from the clinical research centers, from the clinical care centers, and the discovery centers to the biomarkers and biobank and discovery center, as well as to the data coordinating center. All of that will lead to groundbreaking research and innovation. FAIR really had specific responsibilities for the biobanking and biomarker discovery center. As I mentioned in the previous slide, it is imperative that we collaborate with the FAIR data commons and the FCNs, which are supported also by the FAIR patient registry. The mission is to advance public knowledge of the pathophysiology of food allergy, the identification of improved diagnostics and prognostic markers of disease. FAIR had even more specific responsibilities, which for the Biomarker Discovery Center which is leading the implementation and pursuit of key biomarker generations with the FCNs and with clinical research, which includes all elements of biospecimen collection, handling, data curation, and what is needed for site and patient identification. We also were tasked with providing biomarker-related data storage, curation, and analysis infrastructure. Importantly, leading the training and mentorship of the FCN sites to disseminate the knowledge and infrastructure to improve the quality of immune profiling and system biology data gathering across the FCNs. Also establish and maintain the best practices, which we have called SOPs, to complement the existing repositories where needed, and also to allow for data sharing between the FCNs and the FAIR collaborating partners. The FAIR has also tasked with supporting the design, piloting, and implementation of mechanistic and biomarker studies, developing protocols, manuals of operation and procedures, which we talked about, to conduct mechanistic studies to complement the FCN research and patient care, support data management and reporting, and receive and store and distribute banked quality controlled biospecimens, and also grow this research over time. Additional responsibilities were to prepare reports and analysis for presentations and publications, prepare per protocol budgets with the FCNs to have additional monetary support to the biobank itself to conduct mechanistic studies. So all of us will be seeking monetary support for the mechanistic studies and for the discovery centers, and support extramural applications to enhance and complement the financial support of the biobank and the ability of the FCNs to carry out cutting-edge biomarker studies. We're going to do all that hand in hand. And this is where SOS comes in, and I call that Save Our Samples. Because so far there's been very few biobanks and biorepositories for particularly dedicated to food allergy samples. And to do SOS, we don't always have to just line up and be ready to go, but we also have to be ready to battle rough seas. These rough seas are in particular some issues that have been either decided or are waiting to be decided by biobanks and biomarker centers, which is how to navigate the HIPAA requirements, how to navigate informed consent, the subject identifiability, and the question of returning the research results to the research subjects. All that has been tackled by organizations and by the NIH in particular. The National Cancer Institute has developed the Office of Biorepositories and Biospecimens, and the coordinating committee has come up with best practices for biospecimen resources. The RAND Corporation back in a few years ago has come up with the best practices for biospecimen resource for genomic and proteomic era. There is an International Society for Biologic and Environmental Repositories that also contributes best practices. And eventually, a biobank that is abiding by these best practices seeks accreditation from the College of American Pathologists. And our Discover Together Biobank is accredited by CAP, which really includes a view of the biobank procedures every 3 years, and there's also some internal reviews that happen yearly as well to maintain the standards of operation. So after we've battled all these rough seas, we would get to a calmer view here with the biospecimens on one side, the biomarkers on the other, and in the middle we have more decisions to make about who gets access to the biosamples or biospecimens and biomarkers. And we propose that this would happen through an access committee, and we can probably have further discussion tomorrow with the FCNs on this topic, but we have made some recommendations to the FAIR leadership regarding this question. So while we are looking at this very site, I'd like to say that I think that FAIR has been quite visionary in deciding to have a biobank and biomarker discovery center to support the FCNs. We will only know the value of that once it get started, but I know that we're going to appreciate this value as time goes on and as this biobank and the biomarker discoveries start to roll in. I'd like to also say that we are hand in hand with FAIR and have really lined up what I showed you as a long list of cores and researchers to work with the FAIR and the FAIR discovery centers for that purpose. Looking forward to a lot more work to be done. Thank you for your attention.